Dear colleagues, my name is Ekaterina Mironova. I am PhD, head of the Laboratory of Molecular Neuroimmunoendocrinology, St. Petersburg Research Institute of Physiopulmonology, and also senior researcher of Laboratory of Molecular Mechanism of Aging, St. Petersburg Institute of Bioregulation and Gerontology. Today I will tell you about how immunomodulating properties of peptides in vivo. The problem of physiological correction of the immune system function in various pathologies and aging is extremely relevant. People with a history of chronic diseases often experience a severe course of various immunopathological conditions. It has been established that dysfunction of the immune system leads to a decrease in the quality of life, which is expressed in increased fatigue, anxiety, depression, impaired concentration, memory, sleep, and dyspnea. A decrease in the quality of life in such patients often leads to the social maladaptation and decreased ability to work. In this regard, patients need to include in standard therapy additionally safe and effective immunomodulators that normalize cellular and humoral immunity. There are a large number of medicines of various origins and structures that affect the function of the immune system. However, substances of natural origin are of the greatest interest, given their safety and lack of toxic effects. These medicines include peptides isolated primarily from the thymus. It should be noted that the thymus plays a leading role in regulating the immune system, hemostasis, and tissue regeneration. Studying the mechanisms of regulation of the immune system using peptide bioregulators opens up new prospects for the development of immunomodulatory medicines that are highly effective and lack side effects, aimed at normalizing the functions of the immune system. The Vladonix polypeptide complex was isolated from the thymus gland of young animals. Based on the analysis of amino acid composition of Vladonix, the Cristagen tripeptide was designed and then synthesized. Based on the analysis of amino acid composition of Vladonix, the dipeptides Timogen and Vilon were designed and then synthesized. It was found that these short peptides have a wide spectrum of action in different experimental models. A study of the effect of Vladonix was carried out in patients with acquired immunodeficiency conditions, who were administered Vladonix along with standard therapy. After using Vladonix, a tendency toward normalization of the spectrum of lactate dehydrogenase isoenzymes was noted in lymphocytes of the examined patients. This indicates the normalization of intercellular biochemical processes, which are accompanied by the restoration of the functional activity of T lymphocytes in immunodeficiency states. In children with chronic obstructive pulmonary diseases who received Vladonix, the number of leukocytes decreased, while the number of mature T lymphocytes increased, approaching the normal level. Also, after treatment with Vladonix, the content of B lymphocytes normalized. The use of Vladonix led to a decrease in the concentration of cytokines to normal level. It was noted that the use of Vladonix in children with pulmonary diseases normalized the state of the hemostatic system, as well as the content of lymphocytes platelet adhesion. Thus, in children with pulmonary diseases, Vladonix normalized cellular and humoral immunity, which helped prevent the development of a cytokine storm and the occurrence of thromboembolic complications. The use of ladonix and dipeptides Ke for chronic gastroduodenitis in children led to a decrease in the level of pro-inflammatory cytokines in the blood. The restoration of cytokine level in reflection of the stability of immune processes that promote epithelialization of the mucous membrane of the stomach and duodenum. The use of ladonix and dipeptide EW together with standard complex therapy for endometritis in women led to an increase in the absolute and relative number of lymphocytes, the number of T and B lymphocytes, as well as the concentration of IgG. The data obtained indicates the normalizing effect of thymic peptides on the state of adaptive immunity in women with endometritis. In the complex treatment of patients with severe burns, it was shown that the use of ladonix helps to normalize cellular and humoral immunity. In addition, the use of ladonix contributed to the normalization of the production of pro-inflammatory and anti-inflammatory cytokines to the level of healthy donors, which is accompanied by more rapid eliminations of the pathological processes and recovery of patients. Thus, the presented studies revealed the ability of ladonix and its short peptides Ke and EW to effectively restore the function of the immune system in patients with various immunopathological conditions, such as chronic obstructive pulmonary disease, inflammatory pulmonary disease, chronic gastroduodenitis, endometritis, and also in patients with severe burns. Thymic preparations help restore the receptor apparatus of T lymphocytes, normalize biochemical and immunological parameters in various immunopathological conditions. Ladonix and dipeptides Ke and EW have a pronounced inflammatory effects, immunomodulatory effects on the state of cellular and humoral immunity, as well as normalization of the level of pro-inflammatory and anti-inflammatory cytokines, which contributes to a faster recovery of patients. Thank you for your attention.